Case Western Reserve University's Great Thinker Series proudly presents the Origin Science Scholars Program. These lectures are presented by the Institute for the Science of Origins, a partnership of Case Western Reserve University, the Cleveland Museum of Natural History, and IdeaStream, with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. Tonight it is my pleasure to welcome Dr. Greg Forrest. He's the Grant Dahlstrom Distinguished Professor of Mathematics, Biomedical Engineering, Applied Physical Sciences at UNC Chapel Hill. He's also director of the Carolina Center for Interdisciplinary Applied Mathematics. He hails from Louisiana, where he did both his bachelor's and master's degrees at Louisiana State University, New Orleans, after which he went to the University of Arizona for his PhD. Dr. Forrest's research cuts across traditional boundaries between disciplines to beautifully integrate the power of sophisticated mathematical modeling in ways highly specific to the questions people in those very disciplines are asking. This coordination produces scientific insights not obtainable using laboratory work alone. We are very pleased to have him join us tonight to tell us about the virtual lung.
The story about the Virtual Lung Project, it's about 15 years in the making, and it's about people, science, and the translation to medicine and medical biology, and the commitment that can happen through interdisciplinary collaborations. I want to acknowledge those who pay the bills. This is a big group, and the intellectual effort needs to get funded, and our funding comes from the National Science Foundation and National Institutes of Health. Over 15 years. So your government is very good to us. In science, we put the credits first. The people who make this all happen and who have made this bond to help influence lung biology. Rick Boucher has been the director of the Moscow Lung Institute for many, many years, for decades. And what happened was a group of us, primarily it was me, Michael Rubenstein from chemistry, Rich Superfine, who's a microscopist from physics, and Rick Boucher. And we all got together and started discussing the possibility to engineer human lung health. And it's been a long road. I am not a biologist by any means. So I've been continuously re-educating myself. Continuing education is now my passion. And then we succeeded to entrain many, many more people to be able to unravel the mysteries and try to influence medical biology. So a bunch of folks from mathematics, from pharmacology. Tim Elson's dad is a professor at Case Western, by the way. And a whole bunch of important folks who were the originators of the Virtual Lung Project and who convinced us in basic science that this was really something important that we should be working on. The specific collaborators that I'm going to talk about for today are all listed here, and I want to mention two in particular. One is Lingjing Yao, who's a faculty member in mathematics at Case, who's in the audience, and Jacob Witten, who made this real for us. 2 years ago, he was at UMass Amherst as a math physics major. He came to work in my group on this project, and he's now in graduate school at MIT in applied mathematics, and he has cystic fibrosis. So you want to talk about making it real for everyone in the project. This made it real. And Rick Boucher is the one who brought him to work in my group. So the basic question is, what's math got to do with lung biology? That's sort of the nature of this talk, the theme of this talk. And we wanted to put in a plug for next week's lecturer, who is in the audience as well. And so I love this concept of having these public lectures, by the way. We started something similar at UNC when I was what I call a deanlet. I just think it's a great thing to do to continue to interact with the public, to inform you about what's happening. What's math got to do with it? A little play on what's love got to do with it. I was going to download audio from Tina Turner, and I was going to try to do the dance, but I thought better of that. So your lung is a pretty amazing organ, and that's part of what I want to talk about today is what it has evolved to do to pay the price for oxygen exchange, and then what goes wrong when your lung is not functioning properly, and all the different things that can go wrong, and what we're trying to do to be able to assess and correct those things. Mathematical models, most of health and medicine has been driven by statistics. We look for statistical correlations. But there's another aspect of data science, and that is to understand the fundamental mechanisms and build a model that replicates what you observe and then allows you to predict what you don't observe. So those of us in mathematics, and much of what I do now is statistical, we take data, observational, experimental data we invent the experiments to get, and while we are looking for correlates of health and disease, we're at the same time trying to understand the mechanisms of health and disease so that we can predict cures or remedies. So when we make good predictions, that reinforces the theory, but you actually learn a lot more when your models fail. And so one of the things I tell all my students is that confusion is a good thing. That means you're about to understand something. Okay? So when our models break down, we're on the verge of learning something new. Your lung is a fractal structure. I don't want to use buzzwords, but what that means is that if you go down to every scale, it reproduces itself as it branches. That's for a reason. It's an incredible packing mechanism, and in the 25 branches of the lung are there to be able to get down to the 500 million alveoli. Now, you may not like to think about this slide too much, but what comes in with the air that you breathe and how much comes in every day is a function of how much is in the air and how much you breathe every day. So some arithmetic facts. There's about a 1/2 liter in every breath. 16 breaths a minute. How many minutes are there in a day? So how many liters in a day? How many particles come in with every liter of air that you breathe? Particulates, dust, pollen, environmental particulates. So every day, 100 to 200 billion particles come in with the air that you breathe. There's also microbes in every liter, and depending on where you work or what environment you're in, these numbers could be low. So on the order of 1 million to 100 million microbes per day you're breathing, and yet your lungs are probably sterile. And the question is, how could that be? What did nature do to deal with this load that you have to pay for oxygen exchange? Mucus is nature's solution. So my kids all love the fact that they love to tell their friends, they're not kids anymore, they're 20 and 26, that I make a living studying mucus. So an airway cross-section has the tissue with the cells that manufacture the proteins and the molecules that make up the solid structure in mucus. There's a carpet of cilia. Ciliated cells have about 200 cilia per cell. They form a dense carpet of cilia. And above that is a mucus layer I will explain in a little bit more detail. This PCL layer where the cilia bathe is 7 microns thick, and the cilia in full extension are 8 microns, so they stick their head out and engage the mucus in their power stroke. And the idea is they somehow are involved in transporting this mucus layer. This is a stain of 2 different types of mucin molecules. It turns out there's known at least 10 different mucin molecules, and they're in different proportions in every organ of your body. Every organ of your body is protected by mucus. There's a mucus layer on every organ in your body. How does it work? I was encouraged to put some cartoons in this lecture. In every organ of your body, mucus plays a role. It coats your eyes, it coats your nose, it coats your intestines, it keeps your intestines from eating itself due to the acids that are going to digest your food, etc. In the intestines, it's actually a great thing that it hosts all the microbiome that does work for you. Other ways, and here's something that happens when you're ill, which we won't dwell on that. Drink a lot of water. Your body makes 2 liters of mucus every day. You swallow a liter of mucus every day from your lung. And coming back to how does your body deal with this massive load, there's a tennis court of surface area between air and mucus in each of your lungs. So your body is created this fractal structure not just to stack a bunch of alveoli, but at the same time to create a fluid that is going to trap that load, that particulate load. So this is just an illustration of the different, it's color coding the different type of mucin proteins that are in a healthy lung and a healthy mucus in the right proportions to create the gel properties of mucus that both manage the diffusion of the particulates that land in the mucus layer and then allow it to be transported by physiological forces. So I wanted to show you an image from a cell culture of the manufacture of the proteins that replenish the mucus layer. Since it's being transported continuously, it has to be replenished continuously. And here are goblet cells that are bursting to drop packets of mucins. You have to look very closely, and you'll see popping in these goblet cells as they release a payload of mucins. And the picture I showed you back when that was green and red stained, those are the molecules that come out and they phase separate to create the distribution that's required in the lung. My friend Rick Boucher calls mucus nature's flypaper. Everything that lands in this tennis court of surface area as it's transported sticks to the mucus layer. And then it tries to make its way, for example, viruses or bacteria or carcinogenic particles. If they diffuse through that mucus layer and enter the bloodstream or the cells, you've got a problem. So cilia, by the way, are programmed in the womb by the flow of amniotic fluid to have a bias to have a power stroke up toward the trachea. So they have broken symmetry. There's no reason that they would want to bias transport up, but it's programmed in the fetus by the flow of amniotic fluid. We said earlier that mucus is one way to transport. Air drag, your breathing is also asymmetric. Your inhalation, exhalation is biased to transport mucus by air drag. And mucus is believed to transport like an escalator. It is continuously moving due to cilia and air drag or cough. And the game is your lungs are healthy and sterile because it's transporting the mucus faster than the microbes can either diffuse through the mucus barrier, which is only, by the way, 20 to 50 microns thick, sheets of paper thick. And if they don't transport fast enough, then things like bacterial colonies form. So you probably thought, as I did, that the immune system is what keeps your lung healthy, but it's not. The immune system is only there as a backup plan. Most of the time, you don't even have to worry about the immune system because it's cleared. So this system that we have, this lung, is a continuous feedback between mechanics, biochemistry, and biophysics. Just for you to see transport, this is from a sedated mouse and an imaging of flow of the trachea by cilia. You're seeing the mouse breathing, and then we stain some particulates to watch it be transported to get an estimate for transport in a mouse.
This mucus looks like it's a carpet, but it probably has all sorts of other things in it as well.
I will tell you, I'm going to show you next the kinds of model systems we create to create pure mucus by cultures. But the living mucus has full of everything that comes in with breathing. It has dead cells that are also being transported who dump their DNA. And it's full of hundreds of proteins. So it is where I come from, we call that gumbo. It is full of many, many, many different species. So it is truly a complex fluid system.
So there are medications called mucolytics that suppress mucus, and people take them in times of a cold or something. What effect would a mucolytic have on this mechanism that you demonstrated?
This is a public lecture that's going to be broadcast. So I have to be very careful about what I say. But let me say, I don't take any mucolytic agents. There's an interesting story. I don't know how much time I have, but the best thing to improve, especially for a disease like cystic fibrosis, to improve your health is a hypertonic saline solution. And the way they discovered it were a bunch of Australian swimmers who had CF were presenting the disease much later in life. And they finally figured out, well, gee, they're in a saline-rich environment there all the time. And now that is the most scientifically proven method, is to take hypertonic saline if your mucus is hydrated.
Is the composition of mucus the same, males, females, young, old?
Definitely not. There's very little data, what I would call longitudinal comprehensive data, but I'm going to show you what happens during progression if you have disease. And in fact, what happens is you get chemically out of balance. Many things could lead to that, as I'll go through several of the things that could happen, but there's no doubt that the composition evolves. Whether or not we can distinguish different compositions in males and females or youth or whatever, we're involved in the recognition that you really need to know the chemical composition of mucus at the level of detail I'm going to talk about is not so old. It's very recent. So not much of that data was collected because no one was aware that it was relevant and that it could be a key to remedies. There's a lot of raging discussions in the lung biology community, some of whom are in the audience, about what more should you be doing beyond typical known remedies that work, like hypertonic saline, percussive therapy.
Thank you for joining us. You have been watching Dr. Greg Forrest discussing the role of mucus in the functioning of our lungs. For more information on the Origin Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Forrest discusses what can happen when the mucus in our lungs malfunctions. Now, back to the talk.
What can go wrong? Unfortunately, many things can go wrong. And so what we want to do is to go through the things that can go wrong and try to see what we can do about them. I mean, it's not enough to know something's wrong because you have a lung infection and you can't breathe, or you're an asthmatic and you have an attack and you can't breathe. What can we do about it? So I'm going to talk about multiple mechanisms of mucus failure. And what I mean is the failure of the mucus to trap and be transported. The outcome, not the cause, is that you get bacterial infections, biofilms that clog the airways. And then you have a serious problem. You not only can't breathe as well, but you basically have a bloom of bacteria that will grow. So cystic fibrosis is one thing that can go wrong. It's not a whole lot of people worldwide, but it's a very horrific disease, and it's the center that I started this work in. And the problem with cystic fibrosis is an amazing genetic defect in this, what's called the CFTR gene. This protein is what regulates the flow of chloride ions in and out across the membrane. And water follows the ions. So this very simple genetic defect, for the folks who have this, for the people who have this, they're unable to hydrate the mucus layer. So their mucus basically thickens with more of the mucin proteins and other proteins. There is a small number of, so it turns out there are 2,000 known already mutations of the CFTR gene. So cystic fibrosis, like cancer, is not a unique disease. It's a panorama of diseases. But for 3 to 5% of the population, there's a drug that repairs the dysfunction, their particular mutation of that gene, and those folks are living normal lives today. And the goal is, how do you duplicate that for all the other folks who have this genetic mutation of up to 2,000 different kinds? Asthma affects many, many more people worldwide. And asthma is correlated with the fact that you overexpress mucins. So if there's a balance between water, salt, and the proteins in mucus, what's happening is in cystic fibrosis, you're poorly hydrating, so your water is underrepresented, so you have way more weight percent of solids. In asthma, you overexpress the solids, so again, you have an unlevel or an abnormal percentage of solids in your mucus. Another pathology is that your cilia have defects. There's something called primary cilia dyskinesia, and if you've heard about people who's half of the population of folks who have this PCD, their hearts are on the wrong side of their body because their cilia don't, cilia, it turns out, are the mechanism by which you establish a certain symmetry, a polarity. And this disorder is what leads to the inability of your body to set where your heart should be on one side and other types of pathologies. I'm going to show you in a little bit movies of cell cultures that have all of those conditions so that you can visually see a distinct difference when you have these different disorders. Then there's acquired mutations, acquired disorders because of your behavior. If you smoke your entire life, or if you live in a harsh environment, if you happen to be in an environment where you don't know it, that there's nasty stuff in the air. So COPD affects, this is undoubtedly an underestimate, many, many people. And again, this is acquired damage because of lifestyle or conditions that you live in. So what do you do with folks who have lung diseases? Well, you can't exactly, for ethical reasons, go in and start sticking probes down into people's lungs. It's not only painful, but it's risky. You have to anesthetize them, and then you might introduce infections into them. So you can't do this. And then all of the data when I first got into this project, all of the data on induced mucus is all over the place. It's scattered all over the place. And I'm going to show you evidence of reasons that it would be scattered due to just because of what's in your lung due to pathologies. But there's patient compliance. You induce it by giving someone some type of a saline solution, and then they hack something up. How much of it is the saline solution? How much of it is their mucus? Were they telling you the truth about their habits? Who knows? So it's a very imperfect way to test in vivo mucus. So the solution and the transformation in lung biology has been to have ex vivo representations of your lung. Whether you're normal, you have asthma, you have COPD, you have cystic fibrosis, you have primary cilia dyskinesis, what would be great is if we could extract a representation of your lung in a culture. And so I will tell you about cell cultures that were 15 to 20 years old. I could be corrected on that. That's the order of magnitude, in which you replicate genetic mutations in a dish. And I will show you some of those movies in a little bit. And then the most recent transformation in cell biology and in cancer and everything else is that from a single biopsy of your nose, your bronchia, your intestines, your pancreas, your lung, the lung bronchioles, one can reproduce little organoids, little bitty spherical replications of your tissue, and then you can study that, but it's yours. It's your genetic profile, and even possibly your acquired mutation profile. So this is going to transform all of medicine. The prospects for this is, and we're doing it right now with CF pediatric patients. I'll show you some data in a little bit. You take a biopsy from their nose or from their bronchia. You generate multiple organoids that will replicate their tissue, and then you bomb it with every possible drug that you could possibly think of, every cocktail, because we can make hundreds of them from an individual biopsy. And now you can go test what their response is going to be to drugs if it's really an efficacious representation. And that's really is technique. It will happen not long from now. This is what a cell culture looks like from the side. You put nutrients, you take a Gore-Tex membrane, you plant cells from donors. These are unfortunately deceased donors. And those cells differentiate. They decide who wants to be goblet cells. They decide who wants to be ciliated cells. And in about a month, they come alive. And the cilia start coordinating and moving mucus around in the culture. What do you think this looks like, or what this is? That's a top-down view of the cell culture. And this is, credits go to Neil Young. It looks like a hurricane. So we call them mucus hurricanes. That's been transformational. You get sources of mucus. In this culture, the mucus isn't going anywhere like in your lungs, so you have to wash it off every day. That gives you mucus to play with that's replicated from your genetics. You get to observe cilia. You get to observe what happens if I drug it, what happens if I put airflow over it to simulate cough, and you watch the biochemical response. Literally, you can watch that. For example, most people thought decades ago that you cough purely to tug on mucus. But when you cough, it induces a biochemical response, and you dump ATP. That ATP activates something else, and then there's a cascade, and you hydrate. So coughing is not just to tug on mucus. CF patients, by the way, cough their entire life, even in their sleep. Here's an organoid, a little three-dimensional nasosphere. That this organoid has the cilia inside. You can create organoids by technique where the cilia are outside. And you can, again, observe, and they grow much faster, and you can produce them in massive quantities from a biopsy. So this is what's going to happen throughout medical science going forward. Now I want to show you normal cilia and mucus and then pathology. So this is a culture in which you wrap it around so you can conform it to a geometry. And the cilia beat, and you can check to see how they move mucus or whatever type of simulant you put around it. You can wash it off and put other stuff on it. So get a picture in your mind of that normal cilia. They coordinate. It's a miracle. It's a mystery. Not really a mystery, but to be able to explain how they coordinate. Individually, they do what they need to do, beating at 10 to 15 hertz. But then in a little while, within a month in a culture, they coordinate and move mucus around. Okay, in case you didn't see that last time. Here's cystic fibrosis. The cilia are normal. The mucus is not. And there's a qualitative difference in how the cilia are able to engage with that mutated mucus. Here is primary cilia dyskinesia culture, where the cilia have genetic defects and they simply don't know how to beat the way normal cilia do. They're not only ineffective, but they can't even begin to coordinate.
Was the cultured hurricane of mucus movement and organoids, were those in real time, the videos that you showed?
Yes, they were. Yes, they were real time. The cilia beat at 15 hertz, 15 beats per second, so if you can go in and check, and that'll be the way you can tell, but defective cilia don't, and defective mucus will slow down the cilia. In fact, in advanced lung disease, the cilia can't even penetrate the mucus, even if the mucus. So either if the mucus is unhealthy, then normal cilia are unable to penetrate it, or if you have cilia problems, right? So either way it can lead to breakdown. So for example, I'll talk about it in a little bit, but cilia is believed to be the primary transport mechanism. But there's no doubt that air drag works, because you can create, for example, CF mice, their cilia or PCD mice, and you can still see them transport mucus. So air drag definitely works, and there's a reason for that. You have to have secondary mechanisms. And then cough also works, but it works probably for very different reasons than one might have thought originally.
Have any studies been done yet on this new vaping technique as opposed to smoking on the mucus systems?
Absolutely. It is incredible that that was authorized in this country. So in the Marsico Lung Institute, we have significant ongoing studies. You know, the things that happen to your mucus when you inhale stuff like that is pretty incredible. First of all, it stuns your cilia. Literally stuns the cilia, which you can, this is another benefit of cultures, right? You can smoke them. You can vape them and watch what happens. And they induce biochemical responses that may persist for quite some time. So there's a lot of ongoing studies now. It's quite remarkable that this was allowed to take off like it has without the kinds of studies that we normally insist. So yes, those are massively ongoing right now.
I was going to bring to mind for some of us when Jackie Kennedy and President Kennedy lost their child to the hyaline membrane disease. And I remember their physician was a Mary Ellen somebody or other. And I remember hearing a lecture by Mary Ellen who told how much ATP per cilia, and I had that note for a lot of years, but I wondered if you could comment on hyaline membrane disease and how it fits in with your work.
Yeah, I don't know much about the biology of this membrane disease, but I can tell you that I could have gotten into what goes on in an individual cilium. By the way, cilia are replicated across biology. There's a 9+2 architectural structure with 9 beams going all around the periphery, and then 2 beams in the middle that give it a plane of symmetry. And there are 2,000 molecular motors that walk up and down burning ATP to induce the motion of a cilium. We have projects on that as well to try to understand what happens then when you have, for example, PCD, you have a defect in that cilium architecture. And so how does that cascade through the inability of molecular motors to do what they normally would do? That's wide open field of research. It's not at all resolved. But anyway, yeah, I mean, we watch you stress a culture. There's an ATP dump, which we never expected. I'm talking about orders of magnitude dump in the concentration of ATP, and it hyperactivates the cilia. So they're able to power up, or if they were lethargic, they're not lethargic anymore. Water, it induces a biochemical cascade that triggers, that even can bypass defective ion channels. That draws water in, so for a short bit of time you hydrate. Now the cilia can do their thing a lot better. So there's an unbelievable symphony going on when something as trivial as a cough happens. It's really quite remarkable. It's what I mentioned before. It's a feedback system. And you've developed these responses like cough that, again, people thought, I think, for the longest time were just a tug on mucus. But they're doing something far more interesting.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Greg Forrest. Dr. Forrest is director of the Carolina Center for Interdisciplinary Applied Mathematics. In the second part of our talk, we learned what can happen when the mucus in our lungs stops working properly. In our final segment, Dr. Forrest will discuss how mathematical modeling can improve not just our understanding of human organs, like the lung, but the ability of medicine to diagnose and treat disease. Now, back to our talk.
Okay, turn to Patricia and Glenn begged me to put a lot of equations in my talk. And so I thought I'd start with 2 that were in the thesis of this run-of-the-mill scientist named Albert Einstein, who realized, you know, Brown was a botanist who discovered Brownian motion of little particles under a microscope. They're fluctuating. And what Einstein worked out was that you can learn the viscosity of water, honey, molasses, pick it. Mucus will come to next. Mucus is not like water. Just by studying the fluctuations of a particle, a Brownian particle in the fluid. Now, if you just stop and think about that, the fluctuations, they're random. They're water molecules bombing this little probe, this little micron-sized particle, for example. And from those fluctuations, easy formulas, you take the squares of the steps that the particle makes. This is also, you could talk about a drunken soldier who randomly does a walk. And the drunken soldier will get from its original light post proportional to the time that it's drunk and walking. Same thing with the Brownian particle. And just from the slope of this ratio, you get the diffusivity. From the diffusivity, there's another relation that goes back to a famous fluid dynamicist named Stokes, and you immediately know the viscosity of that fluid. It's, you know, for a scientist, this is like in stained glass. It's an incredible property of nature, of this world we live in. Mucus is not water. It's not honey. It's got this big, huge mucin molecules and many other proteins that are a lot bigger than water molecules. So it has a solid structure in it. So it's called a viscoelastic fluid. It's okay. It has, you know, kids know this. You play with mucus, and it's kind of elastic-like, right? But it also flows. So it's viscous and elastic. There is a generalization of Einstein that says I can learn your elastic and your viscous properties by studying a Brownian probe. Another thing that gives you like twice the size of goosebumps. The fluctuations of a probe I stick in are going to tell me what your properties are. Okay? Even more interesting, what's the difference? The difference between viscous fluids is it doesn't matter how you mess with water. The viscosity of water, unless you change the temperature, doesn't change no matter if it's turbulent or laminar or just sitting there or dropping a ball in it. Viscoelastic fluids are very, very different. They have a different viscosity and elasticity at every frequency of forcing. You know this. Your kids or grandkids know this. My grandkids know this. Take ketchup. It is a viscoelastic fluid. You try to get it to flow out of the bottle, it won't come. So what do? You shear it at a shear rate. Now you turn it, and it's got a wholly different lower viscosity, and it flows out. Mucus is the same. So nature has decided to force mucus at 15 hertz of cilia to resonate its properties at that frequency, to force it by tidal breathing on the order of 0.1 to 0.2 hertz, to very low frequencies by gravitational drainage, and then across a wide spectrum is the backup by cough. So mucus is tuned biochemically over evolutionary timescales to have multiple mechanisms to clear it in case the primary one doesn't work. So how can we stick probes like Einstein did in a viscoelastic fluid and learn what its viscous and elastic properties are at all frequencies? It's like doing a combinatorial experiment. Here's the data. Fluorescent little micron-sized probes. You can track probes down to 200 nanometers up to 2, 3, 4, 5 microns. That's the data we collect, and I have teamed with a whole bunch of my statistics, probability, and math friends, including Ling Jing, who is in the audience, to take this data and tell you the difference between healthy mucus, unhealthy mucus across disease progression. And the details of that you probably don't want to hear about tonight. So during disease progression, different diseases, normal mucus weight percent solids, the mucins, the salts, and all the other proteins, in normal is around 2% weight percent solids. In COPD, it gets up to 4, on the order of 4%. In CF, it continues to grow until the patient has so debilitating lung infections they pass away. These are what the paths look like for the same exact bead in 3 different weight percent solids mucus. And you can see that in healthy weight percent mucus, a little bit starting to get ill versus the transition where you would detect symptoms. The mobility is collapsing. That mucus is now having completely different diffusive properties. How do we know by analyzing the data? And just to appease Patricia and Glenn, we have another formula. If that was water, so if we dilute mucus to be 99% water, the slope of this increments from Einstein divided by time would be linear. So this slope versus time would have been a straight line of slope 1. It's never slope 1 in mucus. It's always sublinear. In healthy mucus, it's on the order of 0.8, so the curve bent a little bit. With disease progression, what we observe is that this scaling of the mean square displacement is collapsing. To try to turn that into something that could be predictive for the clinic as a biomarker, we now plotted what the slopes of these mean square displacement curves that Einstein told us to look at versus weight percent solids, and it is eerily linear. We have no idea why this is true. All we can tell you is that it is true for cell culture mucus. So coming back to the question of what's different between cell culture mucus and everything that's in your mucus if you're alive, and is this at all predictive of what happens in patient samples? Okay, that same data, we now use this generalization of what Einstein did to go after what your viscous and elastic properties are versus physiological frequencies and weight percent mucus. So these graphs, I don't want you to, we don't want to labor them. They're telling you that so let's stick at a healthy weight percent mucus. This is your elastic properties, your elasticity, and this is your viscosity, which is different at every frequency. How different? Factor of 50 different from cilia to tidal breathing of air drag. So for some reason, healthy mucus is way more elastic from the perspective of cilia than it is of air drag due to breathing. Now look at the viscous properties at the frequency of cilia versus air drag. It's on the order of 100 times more viscous to cilia than it is to air drag. Air drag thinks it's more fluid-like. Then as you get sick, those properties scale again by up to a factor of 100. So disease progression due to all of the physiological forcing frequencies changed by orders of magnitude. So there's a real biophysical impact of you having more weight percent solids. Okay, this is cell culture mucus, published last year. We are now part of the Australian CFRS project. In Australia, they have authorized the medical community to take all pediatric kids from birth, and they bronchoscope them with sedation and go take samples of their mucus throughout their lifetime, starting when they're born. We have access to this data because our Lung Institute is a partner in this CFRS program, and the folks from Australia are over in our campus all the time now. These are the data from cell culture mucus, healthy down to not so healthy but not truly advanced. And these are pediatrics, so these are up to 5-year-old kids. And all of their data for the metrics that we get from those diffusive microbeads, they're scattering all along it. So it does look like we have very good biomarkers by doing these little experiments and studying the fluctuations of microbeads, and it's correlating with these pediatric patients. That's a big deal to us. Okay, here's a movie. One last little thing. We now want to go in and say, can I zoom in at the scale of cilia engaging mucus and study the forces that an individual cilia applies to the mucus as it engages in that 1 micron tip on the power stroke? It comes back, ducking its head, sticks its head up, okay? What is the force that a cilium exerts on a mucus layer? Interesting question. What's the scale of that force? So to do that, we use atomic force microscopy. And this is a little movie that shows you how to go from millimeters and zoom down to the scale of the tips of cilia engaging mucus. So we're in a spaceship or a microship that's going to take us all the way down to look and probe individual cilia engaging with mucus. So what, and Rich Superfine's one of the Pied Pipers of the Virtual Lung Project, and he's a world-class microscopist. What we do now is we take a microbead and we stick it to a magnetic bead, micron in diameter. We stick it to the tip of a cilium, and then we oppose the power stroke with a magnetic force from the atomic force microscope. And then you tune how much force is needed to interact with and install that cilium, and it's on the order of 60 piconewtons. A piconewton is 10 to the minus 12th of a newton, and you can go look up a newton. I don't have a whole lot of time to talk, but newton's like, you know, how much force is needed to accelerate a kilogram 1 meter per second every second. Now we're down at the scale of cilia, and it's 10 to the minus 12 newtons, okay? Very small forces, very small length scale. So now we take this data, we now create a mucus mimic. This experiment is published in 3 years ago now. We take a magnetic bead that again we can push with an atomic force microscope, and we start pushing it with a force that's on the order of a piconewton. And then we start raising the magnetic force. And up till 10 piconewtons, the bead translates at a steady velocity. It takes a little bit of time to scale up, and then it translates. And below 10 piconewtons, you would see that bead just go on forever as long as you had that level force on. But as soon as you start to get up on the order of a half or a fourth of the stall force of a cilium, that bead no longer behaves that way. It goes through an acceleration phase and then goes at a completely different velocity. And what's happening is that mucus is this highly entangled polymer mesh, cross-linked by a lot of proteins. And the implication is the cilia tip does not just sample the mucus environment. The mucus talks back to the cilia tip because the tip is moving at a force that's stacking all the mucin protein mesh. So it's strain hardening. What is strain hardening good for? Well, you think about an escalator. The mucus in the power stroke is tugging on that escalator and allowed to move it like an escalator, but then it rapidly relaxes so that it doesn't recoil. So this is an insight into what the mechanisms of mucus in a healthy environment are. Okay, I'm just about done now. I wanted to make a pitch for the fact that, first of all, 2 faculty on the Case Mathematics Department were part of this project. Their dissertations came from the Virtual Lung Project, along with 7 other of our PhD students. So this is the flow of translational science and medicine back to basic science. We had to solve many problems mathematically, and there were 9 dissertations, PhD dissertations, generated from this collaboration. And then I'll just stop here.
This lecture is part of the Origins Science Scholars Program of the Institute for the Science of Origins, a partnership of Case Western Reserve University, the Cleveland Museum of Natural History, and IdeaStream. It has been brought to you with the assistance of Case Western Reserve University's College of Arts and Sciences, Siegel Lifelong Learning Program, and MediaVision. For more information on the Origins Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.